everybody. So I will spend a few minutes now introducing DNAlytics and providing a few examples of the different applications we propose in the fields of artificial intelligence, and especially the application of our data science techniques in the healthcare sector. So in a nutshell, DNAlytics was created in 2012 as a spin-off from French-speaking Belgium University in Louvain-la-Neuve, and we still operate from Louvain-la-Neuve. Nowadays, we benefit from a strong team of data science experts, as well as the advices and the environment from a number of industry and biotech veterans advising us on the development of the company. And we develop this activity in a business model which is actually subdivided in two aspects. On the one hand, we operate through service model, so to say consultancy, meaning that we make our expertise available to you guys or to any organization that require the access to data science or machine learning expertise in the framework of their developments. And this is made available, meaning that the outcome remains the property of our partners. And as a second business model, we also industrialize a number of applications to make them available as diagnostics or prognostics tools in a clinical environment. So we productify these results and we sell those products on the market. You will see some examples, of course. As you may imagine from the name of the company, a number of our projects was involving the processing of data, mainly coming from genomics information, DNAlytics. But as a matter of fact, DNA and genomics information is not the only type of material or information we produce and we process in our models. As you can see on this small picture, we process actually a broad variety of input data, be it from conventional clinical biology, clinical observations, patient-reported outcomes, whatever. And I would say specificity of DNAlytics is the capability through our models to integrate multiple types of data into the model. So not only develop a model of one specific data set, but integrate different data sets in a given model to enhance the predictive capabilities coming from the different sources of data. We apply this in essentially three areas. The most obvious is what we call clinical research. So we partner with institutions developing preclinical or clinical technologies, and we help them developing approaches to process their data in a clinical context. That's all the applications related, for example, to the discovery or validation of biomarkers, the classification of populations in clinical studies, or the prognosis attached to specific patients in a clinical environment. Again, you'll see some examples. As a second range of applications, we call it public health applications. It's essentially the extension of those kind of investigations in a broad population, giving the possibility to extract from a given population a number of markers or macro features that help understanding this population. This can serve, for example, to drive policy making in the healthcare sector. And as a third area of applications of our techniques, we use these machine learning approaches in, to support the biomanufacturing or the biopharmaceutical production activities. Interestingly, the same models, the same approaches from data handling can be applied to bring together the different sources of data that are being produced when a pharmaceutical company is producing a drug, and help this company understanding the production processes and enhance these production processes for a better performance or better profitability in their operational activities. We partner with a broad variety of institutions, as a matter of fact, the biotech and pharma companies, as well as the CROs, and typically, as you see on the bottom right, CROs like to work with us when they require approaches or data analytics approaches that go beyond the traditional biostatistics that they typically handle internally. When they do require additional expertise in terms of data science, they would call for that expertise from outside. And we also partner with research institutions, universities, and research laboratories, as well as clinical institutions. It's important to highlight why to call for specific experts in data science, and I think it was already touched by a few of the presentations before me this morning. It's not just a question of pulling together the data and extracting some features. It's also important to be sure that what has been produced is robust, is strong, and can generalize pretty nicely over the population of interest. And this example is, in my opinion, striking because it shows the importance of getting beyond the claims that could be used from a, I would say, marketing perspective, and the importance of understanding what to expect in reality from a model. So in a model, it's not important only to qualify the performance of the model or to talk about the precision of a model, but it's important to define what we call as a precision, and for example, to distinguish the sensitivity, the specificity of what the model can perform. And as you can see from that example, if you just hide these important concepts of sensitivity, specificity, you can have a number of side effects of these models, like in the present example, the capability of producing a large number of false positives, which would, in that case, alarm the population in a way which is just not appropriate for medical application. Why DNAlytics? What is the specificity of DNAlytics as a company? First, I would say that in opposition to the two previous examples that were focusing on what I would call big data, so to say the capability of extracting meaningful information from large quantity of data, in the examples that we're mentioning before, it was imaging information. We like to talk about the processing of fat data. What we call by fat data is the capability of extract intelligence from data sets which are usually not so large. We process the information coming from a low number of individuals, typically a few tens, few hundreds of individuals, typically in a clinical study, for example. But those samples are characterized by a very large number of parameters. And out of those hundreds, thousands of parameters, when we talk about genomic sequencing, for example, our duty is to extract meaningful information, extract the signature, which is relevant for the targeted clinical purpose. And that's a specificity, that's something which calls from very specific types of data science models. We do that on the basis of well-established models and technologies. Typically, we work within the R environment for the experts among you. And we leverage the different data toolboxes and modules that are being developed over the R environment. But we also produce our own toolbox, which is being used again and again over the different models, which helps us being efficient and performing well in these different models, because we come within a new project without any assumption. We just try and evaluate a different given number of models and select the best model appropriate for that specific application, and out of this application we validate the model and we develop it for routine use. And once this has been selected, we can develop this and integrate and encapsulate into an application which is made available to our users. Now a few examples. The first on the left refers to a problematic in pediatric environment related to the need for liver transplants in children. It is well known in that community that a risk factor which relates to the kids requiring a liver transplant is the occurrence of esophageal bleeding. Typically, when esophageal bleeding is being observed in those children, it's a very alarming message indicating that there is a very strong urgency to proceed with the liver transplant. But as you know, the availability of organs for transplantation is pretty scarce in all our countries. So the challenge is to prioritize the kids according to the risk of developing those adverse events. Typically, without any specific rule for prioritization, the kids will proceed into the transplant in a first-in, first-served basis, meaning that they will just queue according to their visits in the doctors, and if there is no specific indication for a risk, well, they will go along that list. In opposition to that, we have developed with the clinicians from Brussels University Hospital with whom we were partnering a methodology which is based over clinical observations as well as blood markers. And from those markers, only 3 markers incorporated into a specific predicting model, we could develop a model capable of predicting the risk of esophageal bleeding. This was developed based on the data available on a retrospective cohort. It was then validated prospectively over an independent cohort and was proven suitable for prioritizing the kids according to the risk of developing esophageal bleeding. And with this tool, now the clinicians has a possibility to put the kids in the right order in the waiting queue for addressing these kids when the organs are available. As a second example of the applications we developed based on our machine learning models is the capability of extracting pathogenic mutations in specific genes. This was developed for a company, a Belgian company called Zentech, which commercialized a number of tests, diagnostics tests, for a number of pathologies. And in that situation, they were investigating blood disorders in kids, and they knew that there were mutations attached to the specific gene which is quoted there. But the question was, which kind of assays to be developed, which mutations to screen specifically for anticipating those, the occurrence of those blood disorders. And by screening a combination of proprietary data sets that they had harvested from their clinical partners, together with publicly available data sets, we could help them outline the mutations that were relevant for predicting that specific mutation. Another interesting example, because it processes over data which are non-quantitative, which are qualitative in those kind of data. This is psychometric data. We've been working with a tool for patient CRO in a project related to the quantification of the placebo response into the patients. That's, the purpose was to try and design a questionnaire, and the analysis tool together with this questionnaire to try and quantify or sort the exposition of patients into the placebo effect. As a matter of fact, there's no way we can exclude this placebo effect. This is a psychometric characteristic of the patient, but by giving the possibility to quantify that characteristic into the patient, then the CRO was able to split or to spread those patients homogeneously along the different arms of the clinical studies, and therefore prevent that characteristic to become a confounding factor into the analysis of the resulting clinical trial. And the other example refers to another traditional data which can be used beyond the traditional clinical use for predictive purpose. In that example, it's for a company, a French-US company developing immunologic treatment against food allergies. The purpose was to see first whether the patients were responding to the drug, and second, whether there was a possibility to predict the level of desensitization to this food allergy. It's a project in which we've developed the model over very traditional markers. These are serum markers that are being measured, that are being measured in conventional clinical biology, but by associating those markers together in a smart way, we've demonstrated the possibility not only to highlight after a few weeks of treatment whether there was something happening within the patient, and second, we are still working with those partners to see whether we can predict the desensitization down the road. The results are not yet published. And I will end up I will go fast over that example because it goes out of the diagnostics or clinical applications. As I was mentioning in the introduction, these tools prove pretty nicely useful also for qualifying or monitoring the performance of the manufacturing process within the biopharmaceutical sector. But I would like to end up with what we call our flagship realization at DNAlytics, which is RemaKit. RemaKit is a precision medicine platform that we have developed together with our clinical partners, and we are now commercializing as a clinical product, as a medical device. The platform is actually able to perform two different specific indications or applications. On the one hand, we are capable with this tool to perform early differential diagnostics, typically for specific arthritis conditions when it's difficult from a clinical perspective to differentiate between rheumatoid arthritis, osteoarthritis, and spondyloarthritis conditions by measuring a number of genomic characteristics within the patients and combining these genomic, to be more specific, transcriptomic markers together with clinical observations. We are capable of performing this differential diagnostics very early in the development of the disease. When I say early, we've demonstrated the capability of anticipating clinical diagnostics by up to 3 years, which is important for the well-being of the patient. And over the same set of data, transcriptomic data combined with clinical observations, we can quantify the activity of a number of metabolic pathways that are known to take place in the development of these rheumatoid conditions. And the next application that we are working on the basis of that platform is to see whether the activity, the understanding or the knowledge over these metabolic pathways can help developing a predictive model that would help selecting the best treatment for rheumatoid arthritis patients. You may know that for that specific indications, there are roughly 10 to 15 different biological drugs available on the market. There is no biomarker, there is no indicator to help choosing the most appropriate drug for the patient. And in up to 40% of the cases, those patients do not respond to the drug they are prescribed in the first line, and they need to switch to a different treatment. This is not only bad for the patient because those treatments come with a high number of side effects, but it's also very bad for the society because these are very, very expensive treatments. So the challenge here is to try and help selecting the right drug for the right patient at first round, and that's what we are working on with our clinical partners. And with this, I will end up my presentation. Thank you.